Our speaker for tonight is Joe Deweese. Joe has a BS in biochemistry from Fried-Hardeman University and a PhD in biochemistry from Vanderbilt University. He taught pharmacy school for 12 years and has spent the last 5 years teaching undergraduates at his alma mater, FHU. He's professor of biochemistry and director of undergraduate research there. He's been researching topoisomerases since 2004 and has published over 40 research articles, review articles, and book chapters. He also co-authored the new book, Our Incredible Human Cell, aimed toward junior high and high school students. He continues to research topoisomerases and is also interested in ancient DNA. He and his wife Liz have been married for over 23 years, and they have two children, and they reside in Henderson, Tennessee, I believe.
Well, thank you, Kirby. Thank you for the invitation, and thank you all for being out there. I know it's not as late for you as it is for me. This is not what the sky looks like tonight here, but this is a night that we saw several months ago, and a friend of mine actually took this particular picture, I believe. But this was a really cool moment for us in Tennessee to get to see some of God's wonders of His creation. Tonight, I want to talk to you about the molecular world and about these amazing things that I like to call life's molecular scissors. As Kirby noted, my wife Liz and I live here in Tennessee with our kids, Lucy and Jed, that you can see there in the top left. Just a little backstory from me. I grew up near St. Louis, about 20 minutes from St. Louis in Illinois. So I'm a Cardinals fan. Grew up there, came to Tennessee to attend Fried-Hardeman University. It's a small private university associated with Churches of Christ in West Tennessee, between Memphis and Nashville. Graduated from there. Liz and I were married a little bit before actually I finished, and then I finished there, and we moved to Middle Tennessee to live near Nashville, where I went to graduate school at Vanderbilt University. I have a PhD in biochemistry from there. And then upon completion of that, I was blessed to have an opportunity to teach biochemistry and pharmacology in a college of pharmacy and also to start my own research lab. And so some of these pictures here are some of my pharmacy students and in the lab that I had at the pharmacy school. And then over here are some of my students from Fried-Hardeman, where I am now. All of these students have now graduated. Most of them are in medical school. In fact, these four, medical school, dental school, medical school, medical school, they're all actually, that whole picture is medical school except for one. Of this crew here, we've got a couple about to start medical school, some that are in med school, pharmacy school, and medical school. So a lot of our students that come through our program looking to get into science and research, many of them are trying to get into med school, pharmacy school, dental school, those types of things. So excited. All these kids are now grown up and big kids, and they're some of them are just starting, some of them are a year or two into their programs. You may not have heard of Fried-Hardeman, but we're kind of a big deal. All right, so what's happening right now for Fried-Hardeman is that our basketball coaches, our men's and women's basketball coaches and their teams are competing in the NAIA basketball championship. In fact, the first round was held today and finishes tomorrow. And we actually we're hosting site for the men's and the women's. Our men's and women's teams are number one in the NAIA, both of them. And the coaches happen to be brother and sister. So that's a pretty cool story. You can look it up, check it out. They both won today, so they'll play again tomorrow, and that'll take them on from there to the round of 16 if they're able to win tomorrow. So just excited about that. That's what's going on here at Fried-Hardeman. Some of the cool stuff that we have going on. In fact, in some of these pictures are down here in the ladies' lines. There's one of my research students is down there on the Lady Lions team, so that's exciting. Okay, so let's talk about terminology for a minute because we throw terms around sometimes, and I want to kind of define and think about. A lot of people call me maybe a biologist or something like that, and I want to kind of define terms. You know, we think about biology. You think about things from maybe the cellular level all the way up to a whole organism, right? That might be what you envision when you think biology. And maybe anatomy would be more specifically, how do the actual organs and bones and everything fit together. Physiology would be the functioning of those going down into maybe the cellular level. Cell biology would kind of be from there down a little bit deeper. Then you get to molecular biology, which would encompass, some overlapping areas there from the actual biochemical level up into the cellular level. Molecular biology really focusing on genes and things like that. And then biochemistry is down here, kind of spanning a range from looking at anything from the molecules themselves all the way up to their functioning at maybe a cellular level. And then chemistry would be, of course, kind of down here in the actual looking at the atoms themselves and studying to understand that. So my realm would be what I would call kind of the biochemistry realm. It's different from microbiology. So some people think, well, you study little stuff, so it's microbiology. No, microbiology is really the study of microbes. So we're talking about bacteria, viruses, things like that. So when you think microbiology, yes, it is small, but I am not a microbiologist. I'm a biochemist. There's another, definition here. Biochemist, someone who solves a problem you didn't know you had in a way you don't understand. See also wizard or magician. So there you go. That's biochemistry. That's a little biochemistry joke for you. So you're welcome. That's free of charge. All right. So this is my parents and my siblings. This is the story of how I got into biochemistry. Okay, because my sister is there next to me. My brother is on the other end, on the other side of my parents. And turns out when my brother was finishing high school and getting ready to go into college, he was diagnosed with cancer. And so it was obviously he's older now, so he survived. Okay, he's a long-term cancer survivor. But my family had no medical training. We had no context for that. And so what does it mean to have cancer? What how do you catch it? Where does it come from, right? We had no context for it. And so that set me on a trajectory, on a journey where I really said, I want to understand cancer. At the same time, as a young man, as I was getting into high school, my brother was about 4 years ahead of me, 3 or 4 years ahead of me, and so as he was finishing high school, I was entering high school, kind of that same era. I also became really interested in apologetics and evidences. So I had this interesting desire to understand science, both from just a perspective of health and cancer, but also from a perspective of understanding God's creation and gaining a deeper understanding. Fast forward a few years, and my siblings and I and our spouses have had a number of kids. In fact, this isn't even all of them anymore. This is a pretty old picture because my kids are pretty little in this one. But I show you this one just because I want to point out one young lady, and this is Lila here. This is my sister's daughter, who at the time of this picture was finishing up treatments for her own cancer. And so what was interesting was we went from an era where we had no healthcare experience, no science experience with my brother and all his treatments to fast forward a few years later. My sister is married to a pharmacist, and that's Lila's dad. And I am a biochemist that studies pharmacology and teaches anti-cancer pharmacology in a college of pharmacy at that time that Lila got sick. And so we had a bunch of different tools at that point to really understand what was going on and to understand Lila's treatment and what that would look like for her and for their family. And so it's just an interesting thing how God brings things full circle and prepares us for things without us realizing it. Because it really, my brother's experience helped prepare all of the rest of us for what my sister and her family would go through with Lila. Thankfully, Lila's still alive, thriving, growing. She and my son are just a few months apart. So Lila and Jed are 11. They'll turn 12 this year. So super excited that Lila too is now essentially a long-term cancer survivor and looks to continue to be growing and healthy. But it does make us motivated, all right, to attack cancer, right? So here's Lila and Jed attacking aliens or something in a video game, right? But it makes us want to what can we do to destroy cancer? And so to understand that, we got to go back down into the molecular world. Okay, so let's think about the molecular world and what's going on in our bodies for a few minutes here, and then we'll kind of circle back around to this idea of how do we target cancers. So if we think about the molecular world, again, we start as an organism, we can zoom in layers, right? We can zoom into organ systems and organs, and then into individual organs, we can zoom into the tissues. And then within those tissues are cells, and within those cells are various compartments like the nucleus here. And then within that are molecules, and then those molecules are made of atoms, right? And so I spend a lot of my time studying kind of the molecular world. What's going on with DNA? What's going on with proteins? How do they work? Those are some of the things that I'm interested in. As we think about it, what does it take for something to be alive? I'm excited you're going to have someone speak about origin of life. That's a huge discussion. But if you just stop and ponder for a minute what it takes for something to be living, that's a fascinating question in and of itself, right? That could take all night and we still wouldn't unpack it all. But a couple of things we do know is one, we know is that cells are the basic unit of life, meaning we are made up of we're told, I haven't counted, but I've been told trillions of cells. I don't think you want to know how many cells in you are bacteria in your gut versus how many are actually you, because there's some thought that maybe the bacteria in your gut almost outnumber your actual cells, but let's not let's not get too grossed out by that. But what do we know about cells? Cells are amazing. In fact, this is a quote from a biochemistry textbook. Now, notice this textbook comes from an evolutionary perspective, right? Assumes no design, assumes no designer, but listen to the language they use. A living cell is a self-contained, self-assembling, self-adjusting, self-perpetuating, constant temperature system of molecules that extracts free energy and raw materials from its environment. While I know that definition sounds a little detached, right? That sounds a little bit distant. It still is amazing that even this textbook has to acknowledge, hey, this is a pretty impressive system, self-contained, self-assembling, self-adjusting, self-perpetuating. It even regulates its own temperature. I mean, this is a really complex system that we're talking about when we get into cells. And then within cells, we have all of these little things we call enzymes, right? Enzymes carry out the jobs, the tasks, the functions in our cells. Enzymes can build up, they can break down or interconvert molecules, changing something from one thing to another. And if we had time, perhaps we could dissect all of the different biochemical pathways that exist in living organisms. We don't have time for that tonight, obviously. But it is a vast array of different pathways used to make all kinds of different fascinating molecules that allow life to exist. Okay, so these pathways you're looking at don't all happen in us, but they happen in nature in different places to build things, to break things down. And what's amazing, and one of the things I point out to my students that I teach at Freed Hardeman is there's so much interconnectedness in these pathways that what is built in one pathway can be broken down and used for something in another pathway. And the interconnectedness is truly fascinating. We need enzymes to do all kinds of things. Copy our DNA, make RNA from our DNA, we call that transcription. Make protein from our RNA, we call that translation. We need enzymes to break things down. We need it to build things up. That could be sugars, lipids, proteins, and even nucleotides. In fact, those are the four primary classes, by the way, of biomolecules in our cells. Sugars or carbohydrates, lipids, fats, proteins, right, made of amino acids, and nucleotides, which make up our DNA and our RNA. Those are the four main categories of molecules. And enzymes are involved in essentially every interchange in those molecules. Whether we're building them up, breaking them down, enzymes are part of that. We need enzymes to make energy because other enzymes need to use that energy. We need it for communication. So the ways that our cells talk to each other and communicate throughout our body is dependent upon enzymes. And we need it for transportation. There are enzymes that help bring things into our cell or kick things out of our cell or export large packets, so to speak, or transport things even within the cell. Everybody's probably seen the YouTube video of the transport proteins walking along the microtubules and things, just fascinating little molecular machines. If you haven't seen that, you can look up later kinesin kinesin motor proteins, and they're absolutely fascinating to think about how those little machines work. So, yeah, all those enzymes and their pathways are just there because of random mutation plus natural selection, right? That's just, it just happens. It's bound to happen. It's inevitable, right? Is what we would perhaps be told by many of our textbooks and other resources is that, well, this is just the natural outflowing of processes and things. Well, okay, perhaps. Would you say the same about this, right? Everybody has one of these probably sitting next to you somewhere. Maybe you're watching on this device. I would not necessarily recommend disassembling it like this because it might be really, really hard to get back together. But what if I told you, well, that thing just happened, right? That assembled itself from some sand and silica, glass particles. I mean, it just, you get a little bit of metal atoms in there and some lithium and some other things, and it just assembled itself, right? You would say, no, that's crazy, because things like this don't assemble themselves, right? This is a highly integrated, highly orchestrated, complex system with many levels of interdependencies, many different manufactured components. This clearly had a design and a designer. There's no doubt, right? So why do we differ then when we talk about it on a molecular level, right? Why do we not see all of these components in a molecular system and say, wow, this has to have an explanation beyond nature, right? What keeps us from going to that? I'd like to talk now for a few minutes about DNA to kind of introduce where I want to head next. As I mentioned earlier, in our cells, we have DNA, and that DNA can be used to make RNA. We call that process transcription. So you hear the word sometimes thrown around called the genome, right? The Human Genome Project or something like that, right? That would be all of the genetic information in an organism. For instance, the human genome would be that for humans. But you can have a bacterial genome or the genome of an apple tree or of corn or of a cow, right? So a genome could be referring to any organism. You just have to say what organism are we talking about? That's all of the DNA in that organism. That information, or at least some of it, gets transcribed into RNA, and all of the RNA in any given organism, we call its transcriptome. Starting to catch on to this here. We have a genome and a transcriptome. RNA, some of that gets translated to make our proteins, which include those enzymes we've been talking about. And all of the proteins made, let's say in a given cell, we would call the proteome. So we have a genome, and from that we get a transcriptome, which is all the RNA molecules that are meaningful to us, and then the proteome, which are the proteins built from those RNA messages. And then proteins, some of them are enzymes and they make other molecules. We would call those metabolites, right? So then we have what we would call a metabolome. So there are people, by the way, that study genomics. There are people who study transcriptomics or proteomics or metabolomics as far as research. So all of these things exist in our modern era as far as ways to study life and the molecules of life. It's pretty fascinating. And then, of course, up here, DNA gets copied. We call that replication, right? So these are kind of fundamental processes that I teach my students about. And what we're learning as we go along here is that this general profile is true, but what we're starting to learn is that some of this RNA that we make can really influence transcription of other sections of the DNA into RNA. And it can also influence which RNAs are translated into protein. And then we have some proteins that are influencing transcription and translation. And we have metabolites that appear to influence transcription and translation. And so the picture seems to be more and more complicated the further we go down the rabbit hole, the more complex this situation is, because it appears that while this general flow of information is true, that it seems that each level can feed back on previous levels and influence what's going on. And that's the current state, I would say, right now in molecular biology and genetics and biochemistry, is we're trying to untangle that web a bit to understand what's going on. And then you also have, by the way, as a side note, sometimes we have enzymes called reverse transcriptases. Several viruses, by the way, express these enzymes, where you can take an RNA sequence and convert it back to DNA. And that's one of the ways that some viruses integrate into our genome, is that they're able to make
At the light.
RNA. So DNA, of course, gets replicated. We talked about that. I'm not going to focus on replication tonight, but replication is important. But I want to point out something that you see on here. So some of these things maybe you've heard of. Maybe you've heard of a polymerase, that is the enzyme that copies, or a helicase that helps unzip the DNA. Maybe you haven't heard of this guy right here, the topoisomerase. And I want to introduce to you why these are so important here in just a minute. First, I want to point out, in the structure of DNA, we often show it as this, right? This little nice little double helix cartoon right here. But the reality is, in our cells, it doesn't exist as just kind of naked DNA like this on its own. Instead, the DNA tends to be wrapped around little proteins we call histones. So these little cylinders here represent proteins that the DNA can wrap around. And it takes about 150 or 160 bases of DNA to wrap around that. Well, then those get compacted together in multiple layers, and ultimately can compact up into those shapes that we think of when you think of chromosomes, all compacted together. It's about 10,000 fold compaction from here to get up to here. It's pretty impressive. In fact, in all of your cells, right, if we took any one of your body cells and we took the DNA out of it and stretched it out end to end, it would be somewhere about 6 feet worth of DNA. So 2 meters of DNA compacted into a space that we measure in, in micrometers, right, or nanometers, right? We're really down on kind of the nanometer scale here. In fact, I'll show you that here in just a minute. So here's a question for you. How do cells do all of this compaction of the DNA without the DNA getting tangled? Okay. We just got through the holiday season. Perhaps some of you put out Christmas lights. Which ones of you, when you wanted to put Christmas lights out on November 1st, because you were an early bird, right? You went up into the attic, or you pulled out the box in the basement where the Christmas lights were, and you looked in it and you realized what? Oh no, we didn't wrap them up. We just threw all the cords in here together, right? And then what happened? You took the box of tangled up Christmas lights, and what did you do with it? You threw it in the trash, and you went down the road to Lowe's or Walmart and you bought some more, right? Is that how it works? Sometimes that's how it works, isn't it? We get frustrated with it. But the reality is, we deal with this idea all the time, and we call it topology. Okay, so I have here a cord wrap. This was my uncle's extension cord reel that was passed along to me when my uncle passed away. It's about 100 feet of cord. It's a lot of cord. Okay, you better keep that organized, or you're going to get really frustrated. I don't even know how much weed eater line this is, but if this weed eater line ever comes off this spool, I might cry because that's a lot. That's a lot of weed eater line. What about this? This is the cable of a pool vacuum. And look what happens to it. What happens here? It's getting all tangled up, right? We call these problems here are topological problems. And they are analogous to problems that happen in our DNA because essentially DNA is like a really long cord or strand. And so any of these kind of winding or tangling problems that we see here can happen in our DNA. Okay. But then you might ask, like my parents did. So my parents did not go to college. They're not graduate level trained. They're not even undergraduate level trained. My dad was a welder. My mom worked for the school systems. And when I was defending my dissertation about 17 years ago, they said, okay, Joe, all this is great, but how do you actually study this stuff? Like, this sounds neat, but how can you study things that you can't see? Because this enzyme, and in fact, the DNA it works on, is effectively invisible to us at a macro level here. So, question before us for the moment is, how do we study what we cannot see? Think about the scale here that we're talking about. So I want to think about this together. We have this. So if we say 1 meter here, everybody maybe is familiar with a millimeter, which is pretty tiny. Centimeters, maybe more. We're more used to seeing a centimeter, which is 2.5 centimeters roughly in an inch. A thousandth of a meter is down here at the millimeter level. Okay. Cells are roughly on the micrometer level. And then as we get down, proteins are down here at kind of this nanometer level. And sub-nanometer, we get down into what we call angstroms, which would be the hundreds of picometers. So that's PICO, picometers. So we're working down here in this zone where, as you think about it, right, we're if this is if a micrometer is a millionth of a meter, right, we're at the billionth of a meter level here. So just think about how tiny that is in terms of space. And the reality is, light microscopy can get us so far, but we're talking about things that are smaller than the wavelength of visible light, right? These are tiny molecules that don't necessarily show up using light microscopy. So we've got to have a way to look at those and study those. This is topoisomerase 2. This structure, I think, happens to be a version of the yeast topoisomerase 2. I think that's where I made this one. You can see half of it is kind of gray, and the other half is colored red, blue, orange, and yellow. And then there's some DNA in green kind of in the middle there. All right, so this enzyme is made of 2 halves that come together. This particular structure is a cartoon rendering based upon what we call X-ray crystallography. So what the researchers did to get this structure is they took topoisomerase enzyme that was purified and they mixed it with salts, and they allowed those salts to the water from that to evaporate and to crystallize. And the crystallized product then was taken and hit with X-rays, and those X-rays diffract into a pattern. And then they use computers to work backward from that pattern to actually solve this structure. It's actually pretty fascinating to think about what they do and how they do to solve these structures. But that's kind of how we got to this image that we're looking at here. There are other methods we can talk about more later if you'd like. But I'd like for you to consider this is how I approach looking at this. As we think about Paul's statement here in Romans 1 and verse 20 that maybe all of you are familiar with, God's invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world in the things that have been made, so they are without excuse. I'm convinced that the topoisomerase and other enzymes like it are made in such a way that we have no excuse, right? We have no excuse but to be able to, understand God's design. So, how do we study the things that we cannot see? It's kind of like, how do we study, God's power? Well, we see his effect, right? We see the effects that he's had in nature. And I think that's how we have to study sometimes the molecular world. We can't see the molecules themselves, but we can see the effects they have on other things. And so that's how I like to think about studying science and studying these things. And so oftentimes we use very simple methods, and I'll show you, like this image here on the screen is a DNA gel, electrophoresis gel. And I'll show you a couple of those to kind of show you a little bit about how we study these things. So let's talk about topoisomerase 2, these reversible molecular scissors. Here's some different views of the enzyme, kind of rotated a little bit so you can see some different angles here. This kind of middle view is the very typical view, but you can kind of see as you rotate it around, it's got It's got different shapes coming in different directions here. It's got almost a heart shape here up top, right? And then when you turn it sideways, it almost looks like it has a mouth down here. I'm going to walk you through and show you what each of these different parts are doing in this enzyme, at least as best we understand, so that you can kind of get some concept. First, though, I've got to teach you this word that I keep saying, so it's not as foreign to you. All right. Topoisomerase. All right. This is your word of the day or the weekend, right? So take this with you through the weekend and share with your friends. The idea is that it comes from the idea of combining the words topology, so topo, isomer, right? So this is the term for a molecule that has the same components but different structural relationship. And then the end, ACE, ASE, refers to the fact that it's an enzyme. So topoisomerase. That's the enzyme that we're talking about. Now, why are topoisomerases needed? Well, there's several reasons, and I want to kind of unpack that for a moment with you. If you took a double-stranded rope and started trying to pull the two strands of that rope apart, what's going to happen if one end of that rope is attached to something? Well, as you start to pull it apart, you notice that as you pull on these ends, what happens to the twists up here? If this is attached to something, it's fixed in space, then this twisting is going to create tension here. And that tension is going to cause these coils to get tighter and tighter and tighter. And ultimately, if that energy of that coiling is allowed to just kind of do what it wants to do, it's going to cause this right here, a supercoil. Maybe that doesn't mean as much to you, but my guess is many of you at one point had a phone. Maybe it was in the kitchen on the wall, and it had that really long coiling cord. Y'all remember that? And occasionally when you went to answer that phone, what would happen? You'd get stuck up against the wall, right? Because you pull the phone off the receiver, you realize the cord is wrapped up so tightly you can't even move it away from, the receiver. And so what did you have to do, right? You had to unplug it, let that cord untwist and untwist and untwist, and then you could plug it back in. That's a topological problem, right? That's topology. And that's happening in our DNA. And so as for instance, we separate these strands of DNA so that we can copy a region of the DNA or perform transcription to make RNA from it. Anytime that happens, we're causing supercoils. And so this has to be dealt with. Another way to view it, on the left here, we have a DNA molecule that is circular, but it's supercoiled. It's all twisted up on itself. The relaxed state would be kind of the open circle. All right, a good analogy for this would be if you have a rubber band nearby, grab a rubber band and twist the ends of it. And what happens? It starts to look like this thing right here. All right, then when you untwist the ends of it, you take it back to what? Back to this circle state. Okay, that's happening in our DNA, except our DNA can't just freely untwist itself. It has to be helped along in the process because sometimes it's not just as simple as untwisting it. It would be more like if we cut the rubber band that we were just talking about, twisted the ends a few times, and then taped it back together. The rubber band would never become untwisted on its own. Does that make sense to everybody? You would have to actually cut it back apart, right? And let the ends twist out and then put it back together. And that's what is happening with the DNA in our cells. So to study this in my lab and in labs like mine that study this protein, very commonly we use small pieces of DNA that are circular, like the one on the screen here. And we can purify these from bacteria. And it turns out that bacteria, typically like E. coli, for instance, which is where we get ours, we just purified some this week, in fact. And it comes out looking like this, all twisted up on itself. And we call that the supercoiled state. And I can then take this DNA and combine it with topoisomerase 2, and it will convert it to this state right here. And how do I know that? Well, I can use something called electrophoresis. So maybe you've heard of electrophoresis. And this is the idea that we can create a little gel. The gel essentially is like Jell-O. Okay, we make it, we melt it in a microwave, heat it up just like you do Jell-O. We let it solidify. We don't eat it because it's not nearly as sweet and perhaps quite toxic depending upon what you put in it. But it has these little holes that we put in it using a comb, and then we can use a pipette to insert our sample into those wells, and then we can apply electric current from negative to positive here. And what happens then is that as the electricity is applied, the DNA will migrate through the gel, and the smallest pieces go to the bottom, and the larger pieces stay closer to the top. So we use electrophoresis routinely in our lab to separate out molecules. So here's what it actually looks like for us. So here, the DNA that is all twisted up like this rubber band here is supercoiled, is going to run faster. And then when our topoisomerase relaxes it to this circle form here, it runs up here at the top. And so what you can see, this is a time course. And so at 30 seconds, a minute, 2.5 minutes, notice what we see here. We kind of see a ladder. And then it keeps going up and keeps going up. And then at 15 minutes, here the DNA is at the top. And what's happening is essentially each one of these bands represents one untwisting of this molecule in the direction of becoming this one. And so you untwist 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's about 11 turns is what it takes to go from the state down here to the state up here. And so the topoisomerase is able to progressively change this to this. And so then we can use that. We published some of this. This is from a paper we did in 2021 from my students back at Lipscomb in Nashville. We made a mutation and we did this and we said, oh, that's not kind of boring. It doesn't really, it looks like the normal one. So then we made some other mutations. Notice this one actually went slower and then this one went faster, right? So we found changes in the protein that we could make that could make it go slower or faster. And so that's part of what my lab has been focused on over the last number of years is examining different parts of the protein, understanding what influences activity, and then thinking through how all of that works. And so these fascinating little machines, what I want to do is kind of show you the steps of that mechanism. I want to show you kind of the steps of that process. How does it actually do that? All right, so this is a diagram, and I'm going to show you a video in a minute that will put all this together. But let me just show you in diagram form for the moment. What we have, here's our little protein model. Okay, so notice the bottom part, the top part, the middle. What's going to happen here is it's going to bind to two pieces of DNA. So there they are bound. The top part can close around that. And then notice what happens here in the middle with the maroon part. This is gold and maroon, by the way. That's our school colors, Freed Hardeman. So go Lions. Everybody cheer them on. So what we see here is you have a double-stranded break in the maroon part. And then we're going to pass the gold piece through that break and then put the maroon back together. And then we can release the gold and then release the maroon. So we've taken two pieces of DNA that are wrapped around each other a little bit or crossing over each other in some way. We've broken one of those segments. We passed another segment through. We put the maroon segment back together, and then we can release them all. Some of the other stuff on here requires magnesium. It requires ATP, right? These are different things that the enzyme needs to do what it does. All right, I'm not gonna, let's see, I'm gonna skip this one for now here. Let me show you then what does this do practically in our cells? Well, we mentioned supercoiling. And so relaxing of supercoiling is one function. Sometimes there are knots in our DNA, like this little knot here. You can remove that. Sometimes two segments of DNA are locked around one another. Maybe these are two separate chromosomes, and the enzyme can unlink them or decatenate them. Now, you notice I have this red and blue here, type 1, type 2. The enzyme I showed you on the last slide was the type 2 enzyme. There's also a type 1, and it can relax DNA, but it tends to not be able to do these other functions, and I'll explain why here in just a minute. Let me go to this animation. And by the way, if you go to YouTube and you search topoisomerase 2 animation, you can watch this thing in full. I'm just going to show you a clip from it, but I worked with the Discovery Institute, the folks up in Seattle, and they had an animator that made this this animation that we're about to watch here, based upon some of the structures I was able to share with them. And I don't take credit for the structures. I just take credit for trying to help organize it. Okay, this is our little protein here. And we're going to zoom in, and I want you to think about two pieces of DNA coming in. Okay, so I skipped back. I wanted to hit pause here. All right. So there's our enzyme. Here's DNA 1, and then here's segment 2. Okay, so there's our two segments. It keeps jumping forward. I'm sorry. Let me get us back to where we were. All right, there's our two pieces of DNA. Notice we're going to close this top gate up here. We're going to break open the middle gate, which then allows the DNA, notice here, is actually attached to the enzyme during that time. It passes the other piece of DNA through, closes that middle gate, and in fact, we think there's a twisting action that occurs up here on the top to keep that all closed, releases the bottom, and then can release the top. Is this not fascinating? If that's not cool, then I'm just going to go to bed because I think we're done, right? All right. So this is really fascinating. This little machine grabs onto that DNA, makes a double-stranded break right there. All right. And notice the ends of the DNA are actually covalently attached. Covalent is a chemistry term, meaning that there's a bond between the DNA and the protein during this moment here. Now that bond gets reversed, okay, so it's going to be broken. But for the moment here, the ends of that broken piece of DNA are attached to either half of the enzyme. This is inherently dangerous because remember, this enzyme's in two halves. So we need those two halves to not fall apart, or we have a double-stranded break, which is bad for our cells. And so it continues on here, though. It stays associated together. It passes the intact piece of DNA through the break. I don't know what the little lightning strike is for whatever reason. The animator added some little lightning bolts in here. But you see the top part's twisting. The bottom part can open up and release. And then the top part can reset. Is that not amazing? Right? This is the invisible attributes, the divine nature of God on display here, that he planned this out so thoughtfully that guess what he said as he designed it? Hey, this DNA is going to get tangled up. I've got a solution for that. And it's this amazing little pair of scissors that can take that DNA, cut it, and then put it back together. Just think, if you had a pair of scissors that could cut your shoelaces and put them back together, that's pretty fascinating. So just to think about how this little thing comes in, it's able to bind to the DNA. It's able to cut one segment of the DNA, pull it apart, pass another piece of DNA through that break. So we'll see here in just a second, and then put everything back together. Fascinating little machine. So these reversible molecular scissors, I was introduced to these as a graduate student long before, by the way, we knew how to make animations like what we're just looking at. I mean, I was studying this thing before we knew what some of these structures clearly looked like, as we have better understanding now. These are cancer drug targets, and the bacterial versions are the targets of antibiotics. So if you've ever taken some oral antibiotics, what are called the fluoroquinolones, for those of you who are in medicine, those target bacterial versions of these enzymes. And then there are cancer drugs that target the human versions of these enzymes. And basically, by blocking their function, you can prevent things like transcription and replication and cell division, which obviously shuts down the growth of cells, which is what's going to basically stop a cancer from being able to spread, right? As if it cannot grow and divide. Now, I've shown you most of the protein, but there's a part of the protein that has not been studied very well yet. So what we're looking at here is the part of the protein that makes a nice, neat crystal structure and looks really pretty on the screen, right? It has these nice ribbons and it looks all cute and pretty. But there's another part of the protein doesn't behave so nicely. And we call that the C-terminal domain, or carboxy. C stands for carboxy terminal domain. And the C-terminus in a protein is just like the back end of the protein, so to speak. And this is like a map of the protein. So the ATPase is up front at the N-terminus, the front end. There's some other domains in the middle. There's a area where it cuts and ligates the DNA here. There's that lower gate that we've been watching open and close. And then there's this part at the very end that we call the C-terminus that is very poorly studied. And here's why. If you look at the structure down here, you see in light gray and dark gray, these parts are analogous to what we were just looking at here. That's basically that, okay? What's this maroon and gold part? Well, the maroon and gold represents the C-terminus. And what does it look like? Well, it doesn't look like nothing, right? It's just a bunch of squiggly lines. This is actually a prediction by the folding algorithm called AlphaFold. For those of you who've ever looked at protein folding, AlphaFold will predict folds for you. And guess what it does with the C-terminus? It doesn't know what to do because it doesn't fold. There are in our cells and throughout our bodies a number of proteins that don't fold into nice, neat shapes. We call this concept disordered proteins. They're disordered in the sense that they don't fold up into nice, neat shapes. Now, that being said, I think they do take on shapes. It's just they take it on in a proper context. And so not all the time do they take up nice, neat shapes. And in fact, when we try to crystallize them, like the experiments that solve these, they're so floppy and flexible that they don't crystallize with the rest of the protein. And so they just look blurry, basically. Well, it turns out one of my colleagues and I, she's a structural biologist down in Texas, a good friend of mine. She is in a situation where she's transitioned from being a scientist to be a stay-at-home mom. And I reached out to her and I said, hey, I've got this project I want your help with. Because I knew she was a fantastic structural biologist and understood how to study protein structure. What I found was that when we use AlphaFold and we put certain changes in the protein, modifications in the protein that are found in nature, and I put that into AlphaFold, AlphaFold actually folds the C-terminus. It no longer just looks like a random squiggle. It's now folded up with the protein. And so we worked on this. We actually published this paper. In fact, let me click on this. So Charisse and I published this paper just last year, and we were able to show that under certain conditions, even AlphaFold folds this into a shape. And for me, this was fascinating because I've been looking at this part of the protein for about 6 or 7 years, and we've never been able to guess what it looks like. And now finally, AlphaFold gave us something to go on. Now, this is a computer prediction, right? This isn't the end of the story. But what was cool is this at least gave us some ideas for what it could look like. And currently, where we're at with this, with my current students, is that we're trying to explore these structures with the idea that we want to make mutations in the protein. And we want to ask if those mutations help us understand how the C-terminus works. The reason this matters, if I can jump back for just a second, this paper right here where we showed this paper from 2021, where we did these mutants, all of these mutations that we did in this 2021 paper were all in the C-terminal domain. And the reason that was a big deal is because people in my field assume that if you change stuff out here, it only messes things up in cells. It's not going to mess things up when you purify that protein because that part isn't needed for those functions. And I was able to show that that's wrong, that in fact, those regions of the protein are important even in a purified system. And so that was a really cool thing that we showed back in 2021. We followed that up. We just published this paper also last year. This has a bunch of my students here. In fact, all of these names other than mine are students. And most of them are all now graduated. I think actually all of these except for Lily, I think, have graduated. So Lily's about to graduate and go to med school this fall. But the rest of these are all out. But they did this study with me where we made some additional mutations. We studied several aspects of how the C-terminus is working. And here's my vision for this and where we're headed with this project is if we can understand how this part of the protein is controlled or how it's doing what it's doing, we can then understand how it influences all of the rest of the protein. And the long-term goal or vision would be, is there a way maybe even to develop a drug that works out here in this flexible region of the C-terminus that then also influences the rest of the activity of the enzyme, where maybe we could shut the enzyme down with a very targeted drug. One of the reasons I want to study this part to do that is there are two versions of these enzymes in our cells. And you'll notice I use this term from time to time, topoisomerase 2 alpha. There's also a beta. And from a cancer perspective, targeting alpha is a better plan than targeting beta. And so I would like to develop a way to target alpha. And it turns out the place that they're the most different is this region here, this C terminus. And so the more we understand what's going on out here, how it works, what it's doing, the more chance we might have at designing a more targeted approach, a more targeted kind of therapeutic. So that's a little bit about what we're doing with topoisomerase 2. It's a really fascinating enzyme, and obviously we could go much, much deeper. I will point this out just in passing here. We focused our time tonight talking about topoisomerase 2 alpha primarily. And of course, I mentioned there's a beta version. But these are really just one type of topoisomerase. Turns out there's at least 5 different families of these enzymes that all have different shapes and different structures and even different mechanisms, right? So the animation I showed you really only applies to this family here. These others all have their own function. And here's kind of one neat little takeaway. All of the enzymes that are type 2 are able to make a double-stranded break. In other words, they can cut both strands of the DNA at the same time. All of the type 1s, guess what they can do? They can only cut one strand of the DNA at a time. And so they have a different mechanism altogether for carrying that out. And so it's fascinating, all of these different families of topoisomerases. In fact, in humans, we express in our cells about 6 different topoisomerases, including topo 2 alpha and beta, as well as 4 others. So it's really fascinating. Even bacteria, E. coli, has multiple topoisomerases in it. In fact, there are even some viruses that encode their own topoisomerases because their genomes are quite large and they have to have that to control the genome. The simplest known bacteria that we know of, even down to genomes that are as small as 500,000 bases, guess what they have? Topoisomerases, right? They have to have, they're all throughout life. And so then the question becomes, of course, well, what do we make of all of this, right? We have these enzymes. They're all over nature. Everybody's got them, from viruses and bacteria, not all viruses, but a few. But bacteria have them, archaea have them, plants, animals, dogs, birds, fish, humans, we all have these topoisomerases. And without them, by the way, without these enzymes, we don't survive. In fact, in our cells, some of our topoisomerases you can knock out, some you cannot. For instance, in mice, when they knock out topo 2 alpha is completely lethal. Nothing survives knocking it out. But if you knock out beta in mice, they will develop maybe for the first few days, and then they will ultimately die of neural developmental failure. So they don't even make it. I don't even think they make it to birth. And so the lack of these enzymes means that those cells cannot control their topology. They can't control how the DNA is being handled or utilized. And so ultimately it spells big time trouble for them. So what I make of this is that God's invisible attributes can be seen in the things that have been made. And what I see in this is I see a God of foresight, right? A God who said, here's a problem and I'm going to solve it. And in fact, get this, not only do we have these amazing enzymes that are solving the problem of topology, Okay, we've got them. Guess what happens if these enzymes get stuck on our DNA? What happens if one of these things malfunctions? There's a whole class of proteins involved in identifying that and in removing topoisomerase when it's stuck on the DNA. So not only did our God of foresight say, I'm going to make sure we've got topoisomerases to keep the DNA from getting tangled up, I'm going to provide the backup system so that if one of these things messes up and gets stuck, we can still remove it, repair the DNA, and move on. How fascinating is that? Right? This is a multi-layered engineering that we're looking at here, where God has said, "Oh, there's this could be a potential problem, and we're also going to solve it." Just totally fascinating to me. If that's not God's eternal power and divine nature on display, I don't know what is. Maybe the aurora behind me, right? That is, but I think we can see it even on the cellular level. And I hope that you can see it there too, because it totally fascinates me. So as I wrap up, I'll mention just a couple things here, and then I'll open it up for questions and discussion. We do at Freed-Hardeman University, we try to get students involved in research. If any of you are kind-hearted and want to partner with us, that QR code takes you to a place where you could donate, and those funds would go directly to research, to funding our summer program, to funding supplies and equipment and reagents and stipends for students, so they're not working for free. If you're interested in that and you have a heart for that, we welcome you to partner with us on that. And you get to help students like this get those opportunities. In fact, this was our crew from this past summer. I don't have a picture of the latest crew yet that's for the upcoming summer, but these were our students from last summer, and most of them were biology and biochemistry students. But you notice the three guys on the right in maroon, those are actually computer science students. This is the first summer we actually also had computer science students working on bioinformatics for us. And that's because one of our latest projects is looking at ancient DNA. And we're trying to extract DNA from ancient sources, various fossils, things like that. I, I'm going to say something that may seem kind of astounding to you, but the letters of DNA that you're looking at right now came out of dinosaur bones. Let that sink in. I can't prove to you yet that this is dinosaur DNA, but we've got some really good evidence that this is real, and we've cross-referenced it with some collaborators, and we're working toward trying to understand sequences like this to see if we can't, demonstrate that this is real. And these informatics students that I just mentioned here on the right helped us kind of get that kick started. I've got three more students with similar training coming to join us this summer. And we're just so excited about some of the things that we get to do at Freed-Hardeman. So I'm super excited about that. I will mention here before I take questions, this book that Kirby mentioned earlier, Our Incredible Human Cell. If you'd like a copy, you can email me at jedeweesebooks@gmail.com or check us out on Facebook, Deweese Family Ministry. I'd be happy to get you a copy. Just let me know. We can, if you have a bunch of copies that you need, we can work out a discount. We have quite a few available and be happy to work that out. It's about 150 pages long, full color, covers a number of different topics about the cell structure, cell organelles, the fundamentals of biochemistry. As was pointed out earlier, Kirby noted it does have some college level information in it, but we also tried to make it very accessible. A lot of diagrams and pictures and images throughout the book to make it visually appealing. The idea is if you've got families that maybe want to have something to supplement their homeschool curriculum, or something that maybe if you're a public school and you want your child to get a different perspective on things they learn about in junior high and high school and even into college, this would be a great resource for that. The late Ed Landry did a lot of the artwork on this and gathered a lot of those images. And so I owe him and Buddy for asking me to be a part of this project. They were already writing this book and well along the way in writing it when they asked me to be a part of it. And so I was able to help kind of get it finished up and clean some things up with them And then they asked me to be a co-author on it, which was a huge honor to me. I'd been praying about an opportunity to do a book like this, and God brought to me Ed and Buddy, and so that was such a blessing to me. So we hope to be able to share that with others. And like I said, we try to, you know, we share this, with whoever wants a copy. Let me know. Money's really not the issue. It's really more about getting it out there and making it available. So. All right, let me stop there and see what questions you all have. Thank you again, for the invitation, Kirby, and for the time that you all have given me. Thankfully, I have not passed out just yet, so I'm still awake and just honored to get to spend some time with you, and would love to open it up for questions or comments.
David Franceschi, I believe. David, go ahead.
Yeah, thanks, Kirby. Do we understand what orchestrates this at a higher level? Is that would that be like in the epigenetics of the cell, or there's got to be kind of like the equivalent of an operating system?
Yeah, that's a great question, and I can't tell you how many times I get asked that.
Yeah.
So I think there's a couple things at play here.
My head.
And this would be in the realm of the fancy word in jargon would be biophysics. Okay. This would be kind of in the biophysical realm, because what we're asking is what happens when these molecules are all twisted and tangled up. And so you got several things that have to happen, right? I think of topoisomerases, and I think the evidence over the last few years, there have been several papers that support this, but I've always kind of thought of topoisomerases as, well, my wife and I live in kind of a rural area, and we have out in the county here, we have a bunch of volunteer fire departments stationed throughout the county, right? With the idea being that you're never too far, hopefully, from a fire truck, right? And I think of the topoisomerases kind of like that. They're stationed along the DNA in different positions with the idea that when tension arises in different areas, they can then respond to that in their immediate area, in their context. So on the one hand, topos are spread all throughout the genome, ready to go. All right, so that's part of the question. The other part is, how do they know when to act? And I'm using "know" here in air quotes, right? Because this is a protein.
That's my question.
It doesn't know anything. All right. There's a couple things that I think help us understand what's going on here. One is that the three-dimensional shape of the topoisomerase accommodates DNA when it's wrapped in a particular direction. Okay, so if you take some rope and you twist it in a right-hand coil direction, it's going to look one way. If you twist it back in a left-hand coil direction, it's going to look another way. And what happens is topoisomerase is designed in such a way that the angle of the active site only fits certain shapes, or let me say it this way: it prefers to fit certain shapes. Okay, and so you have in effect a three-dimensional shape issue here, where it's going to tend to grab this shape but not that shape. And so that's another piece of this. Okay. Another layer is the paper I mentioned that we did in 2021, where we made mutations in the C-terminus. We didn't just pick spots at random. We picked spots that were modified. And the modification in biochemistry is called a phosphorylation. It's where we add a phosphate, which is phosphorus with some oxygens. And those get added on specific spots of the protein. Turns out when that happens, that influences that shape of the C-terminus. And what we know is that certain of those modifications will either allow or prevent the enzyme from binding to DNA. And so there is some instruction, so to speak, that the enzyme receives to either not bind or to bind, right? And so if it has enough phosphorylations on it, then it will associate with DNA. If it doesn't have those, then it will hold off. Right. So there's some of that going in here as well. So there's multiple layers. And you mentioned epigenetics. Well, epigenetics, for the most part, works through the histones, which is the DNA wraps around the histones. The histones have these tails. The tails can get modified with acetyl groups, which is a carbon, methyl groups, things like that. Well, it turns out that topoisomerase also interacts with the tails of histones. And so certain histone tags associate with a part of the protein and in a sense recruit it to a particular location as well. So that is in part involved in this also. Now, it still is a fair question to say, yeah, Joe, that's great, but how does it actually get there? How does it actually know? And those are some of the details that are still being kind of worked out. That's not as clear because, again, we can't see this thing working like and record it on video and say, there it is, right? It's
Right.
at this point, it's still we're having to make inferences based upon biochemistry experiments or biophysics experiments. And so it's not always intuitive, like exactly what's going on there. So that's a
Yeah.
great question. And we're getting more and more of the pieces coming in over time, but it's still, to me, it's still a bit fuzzy because there's still some things where I'm like, yeah, but how does it get there? Or how does it
Yeah.
know to be there? And part of it is in this recruitment. I mentioned that there was an alpha and a beta version. Here's part of the difference. Beta is in essentially all of your cells right now have beta. The cells that make alpha are the ones that are growing and dividing. So if you have, let's say your blood cells are growing and dividing, your skin cells, your hair cells, right? So certain cells are going to express alpha for a phase while they divide, and then they'll stop expressing it. Everything else is just expressing beta. Well, cancer cells rely heavily on alpha because they're growing and dividing. So that's why we would want to target alpha more. Well, there appears to be a really well-controlled mechanism to keep beta on the DNA when it's needed and to keep topo-2 alpha off the DNA until it's needed and then bring it to the DNA. And that involves some of these C-terminal domain modifications that I've been talking about and kind of turning the enzyme on or off. And so there's several layers of information here that appear to be influencing that.
Yeah, thank you, Bob. I know it's an
Yeah,
incredibly
it's a
complex
lot.
question.
It's a
Yeah,
whole
there's a
lot.
lot going.
Thank you,
Thank you.
David. Looks like
Okay,
Kirk.
yeah, Kirk Birchie has a question.
Sure, go ahead, Kirk.
Yeah. So you're predicting some of these shapes with computer programs and folding algorithms.
Mhm.
Are you also able to measure them? Do you have X-ray crystallography facilities somewhere near you that can do that?
All right, great question. Okay, so uh, I mean, there is actually X-ray crystallography not far from us in Illinois, honestly, but
Yeah,
we haven't
exactly.
gone there. Yeah, we haven't gone there because honestly, these computational things that we're seeing, we need to demonstrate them biochemically. So the first step toward that, at least for my group and the way I'm thinking of it, is we're going to make some mutations in the C terminus and look for a functional effect. In other words, if these regions are, let's say, if region A and region B are close to one another, and that has a functional importance, if I change, let's say, something about region B and it no longer associates with A, then maybe that gives me some evidence that that association is important. And so we're going to have to do some really detailed kind of biochemistry like that, which is unfortunately still pretty painfully tedious. I have been trying to leverage AI to help us narrow down some of the best options for that. But it's still going to come down to we've got to design the individual mutants, make the changes, purify the enzymes, and then test for effect. And so that's where we are with this. Unfortunately, we can't really see this in X-ray because we can't get it to the actual protein to stick in those conformations for
Mm.
us. It's still so floppy. And in fact, I saw one lab in Europe, a lady in our field named Valerie Lamour put out a study. This has been now 3 or 4 years ago, but she used what's called cryo-electron microscopy or cryo-EM. And that's where they can take these proteins and freeze them under incredibly cold temperatures and then take thousands upon thousands of pictures of them with an electron microscope. And even using that method, you would think, well, you froze it, it should be, everything should be frozen in place, nothing should be moving. Turns out, even under those conditions, the C terminus takes on so many different conformations, it just looks blurry when they kind of back compute to make the three-dimensional structures. And so unfortunately, we have not yet found conditions that allow us to trap that region in a fixed domain space where we could get a good clear picture of it. So that would be ideal. I think cryo-EM is probably going to be the best bet for it. But I think what it's going to take is us either finding the right thing to bind it to so that it gets stuck in a particular conformation reliably, or maybe make some changes that we can actually get it to stick in a particular position. So yeah, good question, Kirk. That's great.
Okay, I think Kofi's next.
Sure, go ahead, Kofi.
Yeah, so this is very complicated stuff, Joe,
It's
for an
very
engineer,
complicated.
right? So first question, it's a simple question. So roughly how long are these isomerases?
Yeah, so they're down there on kind of that protein scale as we were talking about a minute ago. The idea is we think about measurement, we're down on the level of nanometers.
No, okay, sorry. I meant actually in the number of the number of amino acids long.
Oh, sure, yeah,
Yes.
sure. So Topo2 alpha is 1,531 amino acids, so 1,531. So it's a larger protein. It's not the largest, but there's some quite a bit bigger. But it's a pretty substantial size protein. The typical protein we think of as maybe 300 or 400 amino acids. So this is quite large.
This is quite
Yeah, and then to your point, the part that folds up really nicely and neatly is about the first 1,100 amino acids, and then it's the last about 400 amino acids that is this flexible part. So the flexible part is not a small part. It's actually pretty substantial. It's effectively a whole nother protein, kind of tagged onto the back of this thing, and it's quite large.
And the function that this untangling of DNA, that cutting and the opening up, is that I guess is that set of functions, is that the only thing that this protein does, or do they all do the same thing? They all cut and sort of reattach, or is there some other kind of function that they perform?
Right, great question. So they're all involved in regulating topology. So that could be so that you can do replication or transcription or cell division. So all of those processes need it. But yes, that's what these enzymes are specialized for. Now, saying that, there are some other things that there are some proteins that these enzymes interact with that we don't understand why they should interact. In other words, like these proteins should have nothing to do with each other. So why does topoisomerase 2 interact with this enzyme that let's say moves lipids around? Like why would they interact? So there are some unanswered questions. Is it possible that topoisomerases kind of moonlight with other jobs or roles? I don't know. But the main role appears to be regulation of topology, and it's so critical that without these enzymes, cells die. And so that's enough of a job, I guess, to have. But are there other possible roles? Maybe. But as far as what they cut, they are very specific for DNA. They don't tend to do this to RNA. RNA tends to be single-stranded anyway, but even double-stranded segments, these enzymes don't appear to really pay attention to. So it appears to be limited to DNA and very focused on DNA. And that's what these appear to be really fine-tuned to work with. So good question.
Okay. Thank you.
Doug Weller, I think you're next on the.
Good, thank you. This goes to a question that I've had, and I've actually hunted it down a bit. And you've got 1,500 amino acids, which is parts hooked together. So it's like when robotics, you have the hardware, parts, and then you have some type of software control that drives it. 1,500 bits of information or amino acids is nothing in robotics. And in the cell, we've got millions of these kind of enzyme machines, and they're working in very, very tight interaction with each other. And there's no software, there's no room for software, there's not that much information relatively in the DNA, which is mainly used for the hardware of setting out these 1,500 amino acids in there. So it's all hardware, no control. And so my working thesis is the control in there is just not physically capable with the amount of information in DNA. So it has to be a spiritual operation. In other words, there has to be the Holy Spirit. When it talks about the breath of life for these things to operate in this tight control, there's no software, there's nothing equivalent. when we look at engineering, there's no software available, and there's no processor or anything in these 1,500 amino acids that can either receive the control information or execute on it or store it or interact with each other. There's just not enough. And 1,500 amino acids, and amino acids are just, that's all they are, is they just stick together. And all they do is they have little, adhesive points where they go. And so for these machines to work, there's no control information telling them what to do. And these processes you're talking about, complex, if we were to build something like that out of a robot, you take thousands of lines of code, if not hundreds of thousands, to make it operate precisely the way it does and in coordination with all the other enzyme machines that they have to interact with. So I've just been kind of pushing that idea that maybe there's something more here, not only in the design, but in the actual moment-to-moment function of it that cries out that there's the spirit of like this breath of life that they talk about. If the Lord says, if I take my breath away, everything turns to dust. And in life, you see it because machines don't operate that way. That's just, I wanted to put that out there and see what you thought.
Yeah, that was my question too. That's the exact question. Very, very well said.
Yeah, I've had a number of people suggest that, there might be something to that. I don't want to limit God. Of course, God's capable of operating, so I'm not going to speak for what God chooses to do or not to do as far as interacting with his creation. He's sovereign, and so I'll defer that to him. But I will say we do understand some about how this works, and it doesn't, to me, take any away from God and his majesty or sovereignty, to say that there are some things that we do understand. So I'm going to share my screen just for a second here real quick. And let me click on this one and click this button here. Okay, this is a paper we published in 24. And I want to show you something because I think it kind of illustrates that they're buried. I know you're saying there's 1,500 bits of information in the sequence, but buried in that sequence is other layers of information that help us understand how this thing interacts. Okay, this looks like alphabet soup, right? Does everybody see my alphabet soup here? Okay,
Yes.
It's just a little bit bigger here. All right. Don't focus on the letters. Look at the colors for just a moment. What do you see? We got kind of red. We got some blue. Here's some blue. Red. Blue. Here's some more blue. Here's some more red and some green mixed in there. All right. Do we see these patterns showing up? Red, blue, red, blue. What does that matter? Well, this is the C-terminus, the part that I'm interested in studying. And the red represents negative charges, meaning chemically negative charges, okay? The blue represents positive charges, so lysine and arginine are positively charged amino acids. The green, some of those are S's or T's. Those are sites that can be phosphorylated, meaning if you add a phosphate, they become negative, so they become red, okay? Turns out that this pattern, so what you're looking at here, this is called a logo diagram. In this logo diagram, we generated, there's an online tool for doing it using this thing here called Web Logo. This is, I took 105 different organisms, including humans, and I took their topo 2 alpha sequences and I put them into this web logo. And what it did is it looked at what is the most frequent amino acid at each position. And so ones where it's really tall, like these E's, it's almost always an E in every organism in the list. Okay. And then ones where the letters are shorter or there's multiple letters, that's one that may be a position where it varied between organisms, right? Some had this, some had that. Okay. But what you can see is that there's a very pronounced pattern throughout this thing of positive charges, negative charges, positive charges, negative, and so forth as you go throughout this structure. Okay. I point this out to say that embedded in the sequence of these proteins, in the order of the amino acids and in the chemical nature of these amino acids, God has arranged them in such a way, I believe, that they can carry out these functions and associate with things that they're supposed to associate with and not associate with things that they're not supposed to, based on really incredibly impressive and detailed interaction schemes with these different protein structures. It just blows my mind. And as soon as I made this diagram a couple years ago, I just sat back and stared at it and I said, "This is beautiful, right? This is God's handiwork." Where he says, look, I'm going to hide in here this code, effectively sharing with us how this thing works, right? In part, how the C terminus does what it does is based upon these patterns of charges. Okay, now does that take away from God's power? I don't think so. I think it makes it all the more impressive to note that even within the biochemical realm, God can arrange these amazing things in ways that, wow, once we sit back and look at it, we're like, oh yeah, that's really cool, actually, how that works. And so, does that answer everything? No. To Doug's point, we still don't know exactly how it does some of the things that it does. But I think we're starting to learn in biochemistry and biophysics, we're starting to learn that these charge patterns and some of these things are really critical to influencing how these enzymes and proteins and the DNA are interacting, controlling when and how they interact, how long they interact. I think all of that is in there. So, does the Holy Spirit do it? I have no idea. Okay. I'm not going to confirm nor deny. But what I will say is that from a biochemistry perspective, we are starting to see some clues as to God's fine tuning here. God's the original biochemist, okay? So he planned all this out, and we can see just incredible detail and design in here that helps us get some picture. In fact, we did it with the other protein, topo 2 beta. Look at what we see with topo 2 beta. There's some negative charges, some positive charges, some positive charges, negative charges. In other words, these charge patterns exist, but they're different patterns depending upon which organism we're looking at. And so that's really fascinating too, is that, yeah, this is shared between these enzymes in some sense, but yet it's also different. And I'm convinced that this part of the protein, this C terminus, is how God designed these enzymes to be turned on and off, right? This is the control system, right? If you want to think about the processing unit, here's the interesting thing, Doug. You can cut this part off the protein, and I can stick it in a tube with DNA, and it will do what I talked about earlier. It will relax it, okay? Not as efficiently quite, but it'll do it. It doesn't need this part. So then what's the use of this, right? Why make an extra 400 amino acids worth of protein if you can just chop it off? Well, what we've learned is that 400 amino acids doesn't just do that, right? It plays a role in a bunch of other things. In fact, here's a little map that we made. It has nuclear localization sequences that help it get into the nucleus. It has parts that associate with the histones. That's the CHT part at the end. So it's got things in that C terminus that are really important in a cell, right? That I can't see when I just do my purified enzyme experiments in a test tube. And so you've got to see it in its context in a cell to understand, oh, wait, yeah, each of these little parts plays a role and has a job. So it's really fascinating stuff. So anyway, that was a fun paper. Go ahead.
Just want to mention that's all hardware, and it's all still 1,500 amino acids. It's
Sure.
all hardware,
Yes.
which you can have very sophisticatedly designed hardware, but I'm still looking for the control. Just want to let you know that.
No, that's fair. That's fair. And again, it is sophisticated designed software or hardware, but and I have this conversation, Doug, a lot with folks from. My guess is you're coming from maybe a computer background, engineering background, because we tend to think of things in terms of motherboards and processors and RAM and hard drives and all these things, and the ability of the cell to process data is still something that I think we only barely are beginning to understand. And so, to your point, there's a lot left for us to discover. Now, I'll say this: when I take this enzyme in a tube and I mix it with DNA, and it does this job that we're talking about here, I tell my students, I don't necessarily think that God is intervening, right, in that moment to make the enzyme bind to the DNA and cut it and do all that, right? That's happening in and of itself. Now, I can't prove that God's not interacting with it, right? I have no proof of that. But I think that's innate to the enzyme. It has the ability to do that, and it doesn't necessarily require, intervention in my mind anyway. But again, that doesn't take anything away from the designer of it. He designed the system to work so well on its own. So great question. Thank you, Doug.
I don't see any other hand. Terry Rathman, did
Terry.
you have a question?
Let me be one of the last people to say this is amazing. I never got into these enzymes because I thought they were over the top to begin with.
You're
But
not wrong.
you mentioning that there's 5 different kinds,
Mhm.
and then that means all 5 have to be present at the same time. That's a big problem for evolution.
Yeah, that's a great question. Well, okay, so let's back up and clarify that a little bit. So humans, for instance, we express 6 different versions that are spread across 3 different families. So we spread there's 5 families of topoisomerases, and we have in our cells at least 3 representatives of 3 of those families, which is pretty
Yeah.
fascinating. Now,
Now,
if we go to like E. coli or something, there's about 3 different topoisomerases in E. coli. And we can knock one of those out, but I think the other 2, at least you have to have at least one of the 3, I know, for the E. coli to be able to survive. So to your point, yes, but here's the other interesting thing. These 5 different families are are unrelated to one another in the sense that they would all have to have evolved independently of one another.
yes, there we go.
Okay. So it doesn't matter. You say, well, I got one, we'll just evolve the others. No, they're all independent. You'd have to evolve all of them separately.
Yeah. Your investigation into the charges on the proteins themselves is really good. And you
Thank
already saw, I think, by phosphorylating the DNA.
you.
So when you have charges, you have water solvating.
Yes.
We have to keep water in mind because that might be the mechanism by which the disturbance is checked. Because we know from, you know, um early MRI studies that there's a difference between normal and normal cells, normal and abnormal cells, the T2s, right?
Mhm.
And so that means where's that? I asked, I remember a while back, well, where's that T2? Where is that change really going on?
Mm.
So I'm thinking, if you're working on these charges on the proteins, you got to be thinking of the buddies too, because they may be working together.
That's right. Yeah,
That
no,
would even
that's
make it more amazing to me. Now,
yeah,
one
absolutely.
one final thing. You said there were some covalent bonds going on. Are there also a lot of hydrogen bonding going on in there, like in the isosomes?
There's lots of non-covalent bonds as well. So yeah, hydrogen bonding, what we call electrostatic effects. There's
Yeah.
hydrophobic effects. So yeah, all those are coming into play, in the interaction between the enzyme and the DNA. Yeah, absolutely. Yeah, all of those.
Thank you, Joe. That was
Yeah,
great. Thank
thank
you,
you, Terry.
Kirby, for squeezing me in.
Sure. I see Ray Cowan is next.
Go ahead, Ray.
Yes, can you hear me okay?
Yeah,
Yes.
we got you. Thank you.
Okay, thank you. Yes, so I worked at the Stanford Linear Accelerator Center for many years doing particle physics, of course, along with Kirk, who worked there too. But in recent years, the accelerator has been repurposed for doing what I believe is the world's highest intensity X-ray laser right now. And so I understand that research tool is particularly good at imaging molecular dynamics and things like that in
Mhm.
solution, not just as in a crystalline form,
Sure.
but in much because of the intensity it can image proteins in much less dense configurations. And I was wondering just what, if any, helpfulness that usefulness that such a device would have for your studies.
Oh yeah, that's a great question. Well, that would be pretty amazing. I guess the question I would still have though is even under those conditions, can we get enough information? Can we get the C terminus of enough of these proteins to behave enough alike that you could get it to resolve, right? Because the problem we're having is all the tails are going in different directions. So, the idea of crystallography works because when you crystallize it, everything crystallizes alike, and the stuff that doesn't show up. And so, even in that scenario, you might still have problems with the tails wanting to go their various ways and not behaving. Where I have seen promise on this, though, Ray, is some people doing these what they call atomic force microscopy, and they can measure, for instance, topoisomerase removing a single supercoil from the DNA using these little, and I'm gonna call them forceps because that's what they call them, but they have these basically DNA tethered between these little forceps attached to a little sensor, and they can detect the action of the topoisomerase in real time, which is just fascinating to me. Like it just blows my mind that we can get that, and that's on a single molecule level. Okay, the experiments I showed you earlier are what we might call an ensemble. In other words, it's a whole bunch of proteins and a whole bunch of DNA all together, all in the same thing, and then you see that conglomerate. But in these atomic force experiments, you can get down to the single molecule level and look at what is one enzyme on one piece of DNA, what is it doing? And that is fascinating. So if anybody would like to help us buy one of those, let me know. We'd be grateful for it. But that does sound pretty cool, Ray, for sure.
One follow-up then would be if I guess my question is probably uh, a very basic question, but how dynamic are or how important to the dynamics of the operation of these molecules are changes in shape as they bind onto a site, they do something, they release, maybe they go to somewhere else in the cell and bind onto a different site that has a different shape. Are the dynamics important, or is it more just the sequence of molecules that are connected to one another?
Yeah, that's a great question. Well, of course, all the images I showed you all showed topo as if it's just this static structure. But as Ray's pointing out, I think it's important for us to note enzymes don't sit still like that in real life. They're breathing, they're moving, and by breathing, I don't mean literally respirating, right? But they're jiggling around on a molecular scale, and I think if we assume that what we're looking at is just this static structure, then we're kind of missing the real picture. It's a very dynamic picture, to Ray's point, and it's topo is moving, it's flexing. It does cycle through different conformations, so we have trapped with X-ray crystallography different shapes of the topoisomerase, and that's how we get to this idea that this gate can open and close, another gate can open and close, the top can twist. Those all come from different crystal structures that have been trapped at different states. And so we have that kind of information about it to help us know, okay, this thing is a dynamic scenario. The animation that I showed you all is kind of our best guess of what happens if you put all of those pieces together. And so understand it is not, we didn't take a miniature video camera and go in and zoom in and watch it, right? This is us saying we have this biochemical evidence of these different stages structurally and biochemically. So let's put those pieces together. And that's what we tried to do for that animation. So yeah, I think it is, to your point, Ray, it's very dynamic.
Thank you.
Yeah.
Kwaku, you're next.
Yes, really enjoyed your talk.
Thank you.
Two kind of related questions. What is known about the time scales of motion, and have those actually been measured? And I was curious if you could use NMR, but I realize it's too big. I think Kofi had asked about the size. So it's probably too big to use a solution NMR techniques to look at the motions. But what is known about the motions?
Yeah,
in terms of type scales of the different domains moving relative to each other.
Yeah, so this is happening quite fast. In grad school, I had the opportunity to collaborate with the lab next door to us, and they had an instrument called a rapid quench kinetics instrument. And what that machine does is allows you to take enzyme and DNA and mix them together really, really rapidly, and then add in another reagent that stopped the reaction really, really quickly. And I was able to run experiments with a short of time frame as 5 milliseconds. All right, so 5 thousandths of a second. And that was the shortest that instrument could do. It couldn't go any faster than that. But that's still, way faster than I could do by hand, right? If I was pipetting and mixing things together, I can't work that fast. But what it allowed us to see is what happens kind of at the beginning of the reaction. And what I can say is that this thing is operating, on millisecond timescales, right? So this thing is cutting DNA in terms of a few milliseconds. It can already cut the DNA, which is just fascinating.
That's very fast.
Yeah, very fast. I don't know. I never measured relaxation, so like the whole cycle process from start to finish. But my understanding is it's happening very fast as well. In fact, I can show you our data where we can relax a plasmid in just a few seconds. I mean, it can go very, very quickly. And that's 11 cycles through, all done, all in sequence. So yeah, it's very, very, very fast as far as time scale.
If you don't mind, I want a quick follow up the energy requirements. So I noticed in the table you showed some of the topoisomerases require ATP and magnesium,
Yes.
but some do not.
Correct.
Can you comment on the energy requirements? And I'm particularly interested in, if we go back to evolution, sort of the idea that you have a nucleotide cooperating with a protein biopolymer, and especially if that's required,
Mhm.
how would those have arisen independently and
Right.
cooperate to work together?
Yeah, so that's the chicken or egg concept, right? Which came first? And because you got to have the enzymes to make the ATP, but you need the ATP to power the enzymes to make, to make them. So it is a chicken or egg concept, or what my friends call causal circularity. The product of the process is needed to carry out the process. That's a real challenge. So the ATP, we think in topoisomerase 2, if you think about the rope analogy I gave earlier, where the rope is all twisted up and you pull the strands apart and you get the supercoiling, kind of like the old telephone cords used to do. There's enough energy in those supercoils, in theory, to power uncoiling them, right? So why would topoisomerase need the ATP? So the thought right now, as best I understand in my field, is that the ATP is needed to keep the two halves of the protein from falling apart while it's doing its job. And so the energy of ATP hydrolysis may be what's helping hold that top part together so that you can pass the DNA through the middle and then put the DNA back together that was cut and so forth without it falling apart and falling away from itself. So that's one thought about what the ATP is being used for, because we don't think it actually needs it to power the strand passage process, because we think there's enough energy in the DNA that's all wound up that it would power itself in some sense, right? It wants to get relaxed, but the ATP may be actually helping hold the enzyme together. Now, there are other versions of topoisomerase, some of the type 1 enzymes that we think maybe the ATP is actually powering the process that takes place to alleviate supercoiling. And there's a version of topoisomerase called reverse gyrase, which actually puts supercoils into the DNA. In fact, takes it beyond the normal. And you say, well, why would you want to do that? Well, there are some organisms, for instance, archaea organisms that live in really harsh conditions, like hyperthermophiles live in really hot conditions. And what does heat do to DNA? Well, it tends to denature the DNA. So in those organisms, it appears that they have enzymes that can actively supercoil that DNA, which then actually helps that DNA stay intact and not denature. So how fascinating is that? Right? Not only do you need the topoisomerase to detangle it, but you actually need one to intentionally tangle it so that it doesn't untangle itself. And so that's pretty crazy to think about as well. And again, that's going to be ATP dependent as well, because it's going to require energy to put those supercoils into that structure. So yeah, great question.
Thank you.
Absolutely.
Kirk, you're next.
Yeah, sorry, I'm asking a second, or I'm making a comment here,
Sure.
Joe. You mentioned AFM, or atomic force microscopy, and I don't quite see how that helps you, but there's a company here in the Bay Area that's co-founded by a friend of mine, a church friend, called Molecular Vista. You might take a look at their stuff.
Okay.
He builds AFMs, and he shines an infrared laser through the tip and is able to look at molecular makeup of what so he looks not only at topography, but also at the molecular structure, or molecular distribution of what's underneath there. And if that would help you in some way, get hold of Sung Park, who's one of the co-founders, and he might even be willing to collaborate with you, interested in looking into this in something other than semiconductors.
Yeah. Well, so the way it's used in biochemistry, and now understand, I've never used the method myself. I've just seen it used in papers and seen its utility. But for instance, in my scenario, if we do these changes, for instance, to the C-terminus, what AFM might be able to tell us is, hey, does this enzyme associate more tightly with the DNA or less tightly with the DNA? Is it more efficient in relieving these, topological tangles or not? Those would be the types of questions we potentially could answer with the right tools. Now I say that I'm not an expert in it and have not used it myself, so it may be that I'm suggesting the wrong tool for the job, right? Asking for the flathead when we need a Phillips, but there may be some utility there depending upon if we're able to get to that. Now, some of these things I'm not interested in, pursuing all different types of technologies, because honestly, sometimes we need collaborators that are already experts in these things. So I don't have to figure all this out because I'm working with undergrads, and I don't have grad students, I don't have postdocs, I don't have master students. And undergrads, there's pretty high turnover, right? Because they graduate and they move on. And they don't have a whole lot of time while they're here to focus on some of this stuff. So that's a continual challenge we have. And so, for instance, I've got a collaborator at Vanderbilt in Nashville, and we hope to use, their ability with microscopy to help us answer some questions that we have with some of their technology. Because I'm not going to, be able to afford a $750,000 microscope, nor the ability to properly utilize, a fluorescence microscope of that magnitude. So yeah, requires some good collaborations like you're talking about to get the right technology in place and the right people that know how to use it. So yeah, I'll look into that, and would be happy to talk with anybody about collaborations and where that could go.
Just
I'll put
contact Sung there. Yeah. Oh, what was that?
Let's see. I put my book email in here. My work email I'll put in separately.
Where it is.
You need that, so put that in the chat too. jdeweese@fhu.edu. So Deweese books at Gmail for the books. Deweese at FHU is my work email.
All right. Thanks.
You're welcome. I think Randy had his hand up.
Yeah, Randy, you had your hand up. So
go ahead. I did. I keep going back and forth here. And thank you, Dr. Deweese. I always learn something, and usually it's over the top of my head, and I try to absorb as much as I can, and I appreciate that. But I'm agnostic, and I struggle with faith. Okay, and a lot of these scientific presentations are often based on supporting the design argument for the existence of a Christian God.
Sure.
Okay, and so my question to you would be, if you had a student of yours, or maybe you have a number of students who are Islam or Hinduism who believe that Allah is not in disagreement with you, or Brahma is not in disagreement with you, what would your, and I understand that in a classroom setting that would be an inappropriate discussion, but what do you say to somebody who's, it almost seems to me that the belief, the faith precedes the science for a lot of people of science who have faith. And how would you respond to that? I guess I'm always looking for intelligent people who believe to be able to answer that question.
Yeah. That's an interesting question.
Thank you, by the way.
Yeah. Thank you, Rainey. I appreciate that. You know, obviously, to your point, did I grow up as in a Christian home? Yes, I did. And was I taught creation, as a young child? Yes, absolutely. I did have a crisis of faith in grad school because every paper you read, the language is that of evolution, and it was overwhelming, honestly. I did not have the tools nor the vernacular to understand, you know, all of what I was reading, nor what its implications were. And I looked at that and I look at my Bible and I'd say, wait a second, what do I do with this? And it hit me pretty hard, as a relatively young kid, fairly newlywed, trying to figure things out right in life, to kind of say, wait a second, is all this really true? Or, am I just hoping that it's true? And for me, honestly, one of the things that pulled me through was studying this one enzyme and saying, wait a second, things like this don't just happen. This doesn't just pop into existence from random bits of information. This is really amazing what this thing does in our cells, and it's essential to all life. And so any life form that had to begin had to have these proteins essentially at the beginning, and not just these, but dozens of others. And so the question is, where does that come from? How is that explained by anything other than a designer? And whatever the identity of that designer, right, had to have some designer in my mind. And that pulled me back time and again, because as I sat in talks and I sat and listened to people present their research as I went through grad school and heard talk after talk, day after day, different presentations, different research going on, I just sat in awe, and I realized all these things I'm studying with this one little protein. At Vanderbilt at the time, there were like 400 different labs. I don't even know how many there are today, but there are like 400 different labs, Randy, and they're all studying some tiny, minute aspect of this universe. And really, in that they were really focused on the molecular world, mainly in life, in those labs. And it was very humbling to me because I realized how little I really knew and how little we really know all collectively, right, about any of this, that people can spend their entire lives. I've been looking at this protein since 2004, and there are still many questions I have on it. Like, you're like, well, aren't you done with that, Joe? Like, haven't you moved on by now? And I just keep looking at it, and there's more questions. Like, wait a second, how does it do that? Where does it go for this? How does it function in this way? And so part of my answer, I think, would be that, Randy. But then from another perspective, I would point you to a friend of mine, Mike Houts. Mike just retired from NASA, had a long career with NASA. Mike has been part of the space propulsion system engineering for the mission to Mars, trying to figure out how do we actually send, people ultimately to Mars. And Mike did not grow up like me, did not grow up in a home of faith. In fact, was an atheist through his schooling, has PhD from MIT, very, very intelligent, and had essentially a crisis of faith and realized, wait a second, all this stuff, all these things that I had been thinking about life and about the world, maybe that's not true, right? Maybe there is something greater going on here. And ultimately experienced, a transformation in his life, not in spite of the science, but because of the science. because the more he studied it, the more he looked at it, the more he said, there's got to be something greater here to explain all of this.
What was his last name again, Joe?
Yes, Houts, Houts. Super nice guy, lives in Huntsville, Alabama. And we've gotten to know each other over the last several years. I guess I've known him 10 or 12 years or so. But just a super nice guy and very, very down to earth, even though he's literally a rocket scientist. So, just it really humbled me. So I co-teach a course at our university. It's a Christian university where I teach. And we do, by the way, Randy, have these discussions about faith and about what, questioning things. One of the classes that I get to co-teach is called Bible and Science. And the dean of our Bible department, who actually was a classmate of mine in college, Justin Rogers, he's a Hebrew scholar. And Justin and I co-teach that course together. And we try to ask these questions like, how do we know what we know? Why do we believe what we believe? What if we're wrong? What does that look like? And because we want our students to try to unpack that some while they're there with us, right? So we can walk with them through that, think through that with them. And so I want you to understand, I don't take this lightly or flippantly, because we do have students that are struggling with their faith in our classes. We've got kids who grew up in Christian homes and maybe are not sure what they believe anymore, or are still trying to develop a faith of their own. And so we want to help them navigate that and to look at the options and to weigh, what we understand about our faith and how we understand our faith. And so I respect, and appreciate your question, because I think it is critical, and I hope I didn't come across flippantly or gloss over that. But
not at all.
To me, that this passage in Romans 1 that I mentioned, this idea that we can see God's eternal power and divine nature in the things that have been made. That's what I come back to, and that's where the Topoisomerases takes me. Is that you got to explain this enzyme somehow, right? Where did it come from? How did it just happen to be? And it's just one in a whole toolkit of tools that are all needed by our cells. I mean, we could go on all night and talk about different layers of information, and the engineers in the audience here would look at that and say, "Wow, this is a highly complex, interdependent, system with multiple layers and facets and hierarchies and redundancies." And because that's how it was designed, and it just blows me away the more I study it and the more I teach about it. It's just very humbling. So thank you, Randy. I appreciate your.
Yeah, I appreciate that too. That was a wonderful presentation. So thank you, Dr. Deweese.
Appreciate it.
Well, with that, I think that we've imposed on Joe enough, and he's it's after 11 o'clock for him if I count my Central Time properly, and you're not on. I don't think you're on. You're not on Eastern time. I don't think so.
No, that's correct.
So anyway, but thank you very much, Joe. That just, as far as I'm concerned, a really wonderful presentation. And I have some more questions, but I'm going to spare you those questions for right now. Maybe I'll send you an email or something. But anyway, would you be so kind as to close us with a word of prayer?
I'd be honored. Thank you. Let's pray together. Father, we thank you for this time that you've given us to just spend together and to think about your creation and your world and the amazing molecular things that we can find in it. And we ask for your guidance and your wisdom and help as we consider these things, increase our faith and help us to understand things better and help us to see your power and divine nature through them. Thank you for your love for us and for sending your son for us and for the ways that we experience your love through him and through those around us. Bless those on this call tonight. And I thank you for Kirby and the others that organized this and brought this into being, and for the opportunity to be here with them. Bless us this night. We thank you and love you in Jesus' name. Amen.